Hi, this is Phil Newman from Longevity.Technology, and I'm delighted to say that I'm joined by Professor George Church. George.
It's great to be here. Thank you.
Oh, well, George, I feel like I've seen you quite a lot over these last few weeks. I did actually see you at the excellent Longevity Summit in Dublin, and I was struck by one of the comments that you made in your presentation about having to hit all 12 hallmarks of aging really to have a material effect with a longevity therapeutic. And I just at the back of my mind, I was wondering if that kind of represents really the 12 hallmarks, are they kind of like an operating system for longevity that we need to perhaps start working with?
Uh, an an operating. Oh, I think it was 10, but it's flexible. They're a complex set of biochemical pathways, regulatory pathways, and the hallmarks are typically in terms of phenotypes. But um, what exactly do you mean by an operating
Well, I
system?
guess that what I'm saying is that a lot of people are perhaps working on one or two of them,
Yes,
and that of course then means that they are kind of siloed in the area that they're working on.
right.
And when I thought about your comment, I was thinking that really it sort of defines the fact that really to have that systemic approach and really having to pick off all 10 or 12, however you count it, is that really identifying like a sort of an operating system or a kind of
Uh,
methodology underneath longevity that we really need to work towards?
Yeah, I think it does. I mean, I think there's been a tendency for wishful thinking that it's some simple food or drug, single drug that was already approved for something else. I share that optimism, but I think we need to work on, you know, uh, plan for the worst and hope for the best, and we have the tools to do it now. I mean, it suddenly it's really been an exponential experience of the progress in making both analytic and synthetic biology tools. Um, and so, yes, an operating system is a good metaphor. I haven't heard it before, where you really have to deal with every aspect. It does you little good to have the part of the operating system that handles disk drives if you don't have the one that handles memory and keyboard and all the rest. So and if and in with the um, with age-related diseases, if you get one pathway or two or even half of them, um, it's quite likely that will only buy you a few years. And we've already bought, you know, maybe half a lifetime over the last century or so by um, working on infectious diseases so well and a few other aspects of it. But to really get it, I think now we're talking about um, almost turning ourselves into a different species. Like the difference between a mouse and a bowhead whale is the difference between living 2 years and living 200, and it's not because of what they're eating or how much exercise they're getting. It's a fundamental operating system difference.
Yeah, well, that's great. Well, I guess the operating system metaphor is one that I'm becoming a bit fixated on. But another thing that I'm actually becoming a bit fixated on personally is Wegovy or Ozempic. Novo Nordisk's semaglutide, and the fact that obviously it's an interesting drug because it supports diabetes related outcomes. It has got cardiovascular benefits, and I did see a study from 2021 that was demonstrating that it has neuroprotective benefits in mice. And I wrote an article last week just saying, is actually this a longevity drug? And I'd be interested in your thoughts, George. Just whether it's on that as a particular drug or a category of drug that perhaps picks off two or three aging diseases. Would that qualify as a longevity drug, or do we need to have a rejuvenation, a systemic rejuvenation therapy that really means true longevity?
Well, I think it's challenging to talk about longevity until you've done some kind of preclinical or clinical trial that shows longevity. And that's one of the reasons I think my team and possibly others um, prefer to focus on the aging reversal or the reversal of multiple age-related diseases at once.
Right.
Nevertheless, you could. I don't know what the status is of studying this in shorter-lived mammals like mice, but that can be somewhat suggestive. Um, but it takes a long time. I mean, we've done it now for 2 gene therapies, and we could go into why I put gene therapies in a separate category, but I think they're very promising and they're very powerful, essentially changing some of our species specific programming in a way that's hard to do with the drugs. Not, I won't say impossible. It's just we have more precise and more levers to move with gene therapy.
Yeah, interesting.
Ozempic, I don't think they've gotten to a point where there's a Kaplan-Meier plot, but even if they have, I mean, all the progress we've seen so far, it's usually a few percentage points, and that's not um, you know that's not what most people would call a longevity drug. I mean, it has an impact on the lifespan. That's true,
Hmm.
but anyway, it's very hopeful. I mean, I hope that the collection of know-how on things like that will eventually. It may turn out that to get it longevity, we have get it all of the hallmarks. You may need a drug for each. Some drugs may knock off 2 at once. Um, some may just extend them by a bit, but if you get all of them and you extend them, and there's some multiplicative factor, we can get it. So it may be. I think it's very likely this polypharmacy we're looking at is something where you have multiple drugs, just like you do for antivirals, antibacterial, and anti-cancers. It's not unusual to have 5 drugs at once. I think this may be one where we're talking about 20 or yeah. Anyway.
Well, I mean, I guess it's interesting when you're talking about perhaps bringing in those multiple therapeutics together, and of course, there are so many things, so many attributes to the science of longevity at the moment. And I'm minded of the Gartner hype cycle, where you go from a technology trigger to the peak of inflated expectations and the trough of disillusionment and back up again. And with your view, George, on so many of the aspects of longevity. Maybe just ask you first of all, what do you think is approaching peak hype now in terms of expectation, like cellular reprogramming, for example? I mean, what's not reached its peak potential yet, do you think?
Well, yeah, I mean, cellular reprogramming is an interesting one because it clearly achieves most of what you would talk about in terms of at setting back the clock, all the clocks, and producing immortal cells
Yeah.
that are basically embryonic forever.
Right.
Um, and partial reprogramming works as well. So that's very promising. But I
Hmm.
know of only one actual longevity study using the Yamanaka factors or a subset of them, which was published in a preprint from Rejuvenate Bio, where the delivery was via AAV. So, I mean, almost by definition, the Yamanaka factors themselves are either protein or gene therapy. Now, there might be a way that you can stimulate that with small molecules, but anyway, that has been tested,
Right.
and it does shift the Kaplan-Meier plot over to the right um, by a significant amount, and and it can be applied at a very late age. It was applied in that case of that preprint at the at 130 weeks or something like that. It was at the point at which half of the mice were half of these healthy, well cared for mice were dying of old age. So
Hmm.
that's in a way that's really the sort of thing that we want. We don't want to have to be taking it um, from prenatal onward
Yeah.
because most
Yeah.
of us are well beyond that point. Uh, so anyway, it's hopeful, but again, it's a small percentage, and that's with our most powerful drug that looks really great in vitro on cells.
Yeah.
But so far, and now part of that's delivery. Um, so we want to get it. You have to get it to a large number of cells, or to a large number of the right cells, and maybe that wasn't done yet. I think it's too early to say something's. In fact, it's always too early to say something's
Of
impossible.
course. Yeah.
Uh, yeah.
And any therapeutic approaches, George, that you feel have kind of gone over that peak and are maybe struggling a bit to prove their efficacy in the field of longevity?
Uh, well, I think it's unfair to say they're past their peak because you never know when one of them will be part of a combined therapy.
Hmm.
Um, I mean, certainly, things like metformin could be part of a combined strategy, even though by itself it's had it's had a lot of chances already. It's had many
Yeah.
shots on. It's not like it's been deprived of clinical trials because it had one established win, which is diabetes, and so
Yeah.
as a consequence, you get a lot of. That's how we've even found out that it had uses beyond diabetes. So I wouldn't characterize it as past its hype cycle. It's hanging in there.
Yeah. Yeah. And I guess that, of course, this whole field is really driven by the fact that biological age is becoming something that is moving into public consciousness, and of course, there are tools that are measuring biological age. Not necessarily all on the same level. They're giving everybody something to push against, but they're not all reading the same values at some point.
Right.
So, what are your thoughts on getting to an aging biomarker or a biological biomarker that we'd all settle on?
Well, I tell you my slightly jaded, cynical view of biomarkers. I
Yeah.
mean, I've my research has benefited from them tremendously over the years, um, and in fact, my PhD thesis was at least half about methylation of DNA, which is arguably one of the likely candidates for getting consensus. The problem with biomarkers is that you can get it right and still be wrong. That is to say, it could be highly correlated, and DNA methylation and transcriptional clocks are extraordinarily correlated. Maybe an error of 2 year standard error of the mean or something. But uh, you could imagine a drug which you've kind of overtrained. It hits every single methyl site that matters. Maybe, for example, making a CRISPR methylase or demethylase that goes in and edits each of those things. And in the end, you're not any different because those aren't causative; they're somehow correlative. Um, the same thing goes for something like statins, which at first were developed entirely going after the biomarker cholesterol, which most of us think of as the real thing, but have to keep in mind that it is a biomarker, and the real thing is youthful, healthy health span. So um, actually a tiny fraction of the people, maybe a percent or two of the people that are taking statins are actually getting their life extended. Some of them are getting um, some unintended negative consequences. Some of them are just neutral.
Hmm.
Um, but it has recently, there have actually been some longevity studies, which, so for at least some people, so some of these age-related things are going to be personalized medicine, and hopefully a lot of them are going to be pretty generic. But biomarkers, I think, have a role certainly in research. But the best assay that I can think of is something that reverses um, an actual disease of aging,
Hmm.
one or more, and you can get that's easy to get past the FDA. And if it has a broader spectrum impact than that, if it actually gets at the core of aging, but you get it approved based on some cardiovascular disease reversal, that's that's a real win.
George, maybe just if we could just change subject matter a little bit to some of these. You have an amazing reach over, prolific as it were, over many startups that are in this space. Some that are outside, like Arctic elephants, but obviously RNA targeting, antibodies, enzymes, and so on. So, what are you looking for when you get involved in these startups? Because of course, it's having George Church on your advisory panel is a good thing to have, but obviously it puts more burden on you as well. So how do you make those choices?
Well, oh, yeah, that's a good question. I try not to be too frugal. If there's something I can do to genuinely help, I don't say yes to everything. In particular, something that could be a reputational hit, something that's flaky.
Yeah,
Uh, but if I see something that other people don't see, I'm not embarrassed to put my name and more importantly, my sweat equity into it. Um, uh, the other category that I will reject, in addition to things that are, just not ready or not not right, is something that just where I can't contribute. So it could be perfectly valid science, but I just don't see that I can contribute. So, but yeah, I'm happy to do it. A lot of them come from postdoctoral fellows from my lab, so I already know we're on the same wavelength. We share the same interest in fact-based um, clinical trials, not just anecdotes, and uh, and so it's pretty easy for me to assess those. A little bit harder when somebody comes out of left field who didn't train, but even those, there've been some interesting um, opportunities.
absolutely, I'm sure. And I guess that in relation to that, what are you working on yourself, George? That's exciting, gets you out of bed and gets you into the lab in the mornings.
Well, a lot of things. Let's see. You've mentioned a couple briefly. I guess one that really gets it, that takes polypharmacy all the way to the extreme, is we've been working on writing genomes. That is to say, not just editing them, but more precisely, writing them. I mean, it's interesting that people got all excited about our CRISPR,
Yeah.
but I was jaded even before the paper came out in 2013, because it seemed like a step backwards. I mean, it was nice for researchers because it was rapid prototyping at its finest, but
Yeah.
in terms of precision of editing, it was a step backwards. Capecchi and Smithies got a Nobel Prize for very precise, pretty big edits, things that we're just now getting back to. Um, Kilobase size corrections, uh, while CRISPR was what I called genome vandalism, where it would just make a mess at your favorite target, which was good if you wanted to get rid of something, but it was hard if you wanted to do something more nuanced or additive. Um, and there have been lots of improvements on CRISPR. They're still just barely incrementally getting back to what we really want, which is arbitrary editing. Anyway, I think we're getting to the point where we not only can do that, but we can edit the whole genome. So if there's lots of changes you need to make, that's possible. And so, for example, when we're making organ transplant, when we're thinking about ways of doing that, we turn to a pig, and we have a paper coming out soon where we've made 69 edits, which is a one up from the 42 that we've already published, and preclinical trials on primates that look like we're getting up to 2 years persistence of the organs, and that's using organ donors that are designed for humans. And it's much harder to do primate trials, ironically, than human,
Oh,
because
really? That's interesting.
all sorts of, I mean, you don't have to worry about compliance with their meds because you could force that on them. But everything else, that they'll rip out sutures. They'll, if you let them, when they do get sick with humans, you will use more extreme measures to keep them alive. And so just also the biochemistry is slightly different. Anyway, so getting 2 years is well beyond what most what the FDA is requiring for preliminary results. So, and anyway, so put those two together. If you could rewrite a genome, more than just satisfying the crucial crisis that we have in lack of organs. So many people dying while they're waiting in line, or having medical problems while they're waiting in line for years. More important than that is the possibility of having enhanced organs.
Right.
And by
Enhanced
organs,
organs.
and what I'm and organs that are resistant to human pathogens. So it
Right.
turns out a huge fraction of human pathogens are human specific. They may have come in from an animal at one point, but it's a rare event, and most of them you get eliminated them from human, and they're gone, they're extinct. So pigs are resistant to a lot of human pathogens. They could be made resistant to even more. So I'm kind of listing things that in a narrative form that get me up in the morning, which is what you asked for. We've shown that we can make at least one species resistant to all viruses, sort of a
Wow.
priori, by just changing just one codon, two codon switch from serine to leucine. That that makes them resistant to even viruses we never characterized before. So maybe we could do that in pigs to make them really resistant. We can make them, since we know how to make animals more senescent resistant, cancer resistant in the germline, can't do that in human, but we can do that in pigs that then contribute to human. And it's not just about organs. It's like, one of the most common transplants is hematopoietic stem cells, which have impact throughout the body. They're found in every organ. That would be a good target. And so something that would be initially used for dealing with a serious disease might eventually be used for something that's good for everyone.
Yeah, that sounds fascinating. So it does sound worth getting out of bed for as well.
Yeah. Well, so that was a cluster of things. That was multiplex editing, writing genomes where you can do the whole genome, um, xenotransplantation from pigs, and then an enhancement for diseases, senescence, and cancer, among others.
Well, it sounds fascinating. And George, we've obviously got RAADfest coming up, and you're going to be one of the presenters there. So what will you be talking about at the show?
Many of the topics we've touched upon today, but I'll be showing data rather than just interviewing. Yeah, and I'll drill down on especially the last topics of what it is that's exciting. Some unpublished work. It'll be good.
Yeah, sounds like fun. Well, George, thanks so much for joining us today, and it's been fascinating. And best of luck at RAADfest.
Thank you, Phil. Looking forward to it.